Genes are coded in your DNA and ultimately give rise to all the traits that make you, you. However, if we do not tightly control which genes are turned on and off, disastrous effects can occur, like cancer or death. This video will cover all the ways gene expression are regulated in the nucleus. First, let's review the parts of the gene. You have to start with the promoter, which is a series of nucleotides, typically TA, that bind to RNA polymerase or other transcription factors that then recruit RNA polymerase in order for a gene to be transcribed into mRNA. Transcription is the first step in a process that ultimately creates a protein that is encoded by the gene. Then you have the gene's coding region, made of exons, which code for segments of the protein. You also have introns, which are segments that are then edited out of the mRNA. How do you remember the difference between the two? Exons are expressed and introns are the junk DNA in between the important parts. Introns are not actually junk. They can be very important for gene regulation, and we'll get into that a little later. Finally, in the gene, you have a terminating signal that signals to RNA polymerase to stop transcribing and to remove itself from the gene. Promoters are all not the same. Some can bind very strongly to RNA polymerase and the transcription factors, while others are weaker and require some help. The first type of gene regulation we'll look into is increasing gene expression. Enhancers are elements that are either upstream of the gene, close by, or even on a completely different chromosome. They're hard to locate because they're physical structures and they're less likely to have a known sequence of nucleotides we can look for. They physically make it easier for transcription factor proteins to bind to. These transcription factor proteins are very good at recruiting RNA polymerase to bind to a gene's promoter. In summary, enhancers make it easier for RNA polymerase to bind to the promoter and therefore make more mRNA copies of the gene in question. Our research goal in the STANX project is to ultimately trap and characterize different enhancers found in Drosophila melanogaster's genome. The second type of gene regulation we will look into is repression, or decreasing gene expression. Common repressors include methyl groups directly attached to nucleotides or histone proteins that hide or make promoters inaccessible to RNA polymerase. Some proteins can also attach to the promoter site so RNA polymerase cannot, therefore making it unlikely the gene is transcribed. The most famous example of this is the lac operon, which produces lactase to digest lactose, the milk sugar. When glucose is present, there's no need for lactase, so the LAC-I repressor protein attaches to the lac operon promoter to prevent the expression of lactase. When there is more lactose present, lactose binds to the repressor protein so it falls off, and RNA polymerase can then transcribe the gene and create the lactase mRNA. Another form of gene regulation is known as alternative splicing. Let's review an important concept about mRNA before it leaves the nucleus in order to understand alternative splicing. See, mRNA, when it is transcribed first, is known as pre-mRNA. It has to be modified before it can be translated into an amino acid chain. This means that it needs to get a 5' cap and a polyadenine tail, but it also needs to have its introns excised or removed. Then its exons are joined together. Removal of introns and joining of exons is known as splicing. The mRNA is now considered fully matured mRNA and is ready to leave the nucleus. However, some pre-mRNAs can have alternative ways to organize the exons and remove the introns, which is known as alternative splicing. A famous example comes from the discam gene in Drosophila, whose product guides nerve cells to their destination before embryonic development. A remarkable 38,000 different mature mRNAs can be made from this one gene. Alternative splicing explains how complex organisms like ourselves can arise from a mere 20,000 genes found in our genome. Genes are important, but without ways to regulate their expression, they would not come to life.